Staphylococcus hyicus is a seldom mentioned bacterial organism which holds great importance in the field of veterinary medicine and even branches into human medicine. S. hyicus is of the phylum Bacillota, class Bacilli, order Bacillales, family Staphylococcaceae, genus Staphylococcus, and species Hyicus. The genus name means bunch of grape-like cocci, which correlates to the appearance of cocci-clustered colonies under a microscope, while the species epithet means of pig, relating to the bacteria's susceptibility to infect pigs. Closely related to S. hyicus is S. chromogenus, which can be isolated from mastitis in cows, and S. scleriferi, which causes epidermal and ear infections in dogs. These will become more relevant later on. When cultured on bovine blood agar, S. hyicus colonies present as white and opaque cocci that are 1 to 3 millimeters in diameter and do not lyse the red blood cells in the media. It is gram-positive and facultatively anaerobic, meaning that it prefers to grow with oxygen, but it is capable of anaerobic growth. The bacteria is catalase positive, meaning it has the catalase enzyme able to convert hydrogen peroxide into oxygen and water. However, it is oxidase negative, meaning that its terminal enzyme in the respiratory pathway is not cytochrome C oxidase. Furthermore, S. hyicus can ferment D-glucose, lactose, sucrose, trehalose, glycerol, and mannose during acid formation. The genome sequence of Staphylococcus hyicus indicates that the size is 2,472,129 base pairs with 1 linear chromosome and 0 plasmids. Staphylococcus hyicus is special as it is one of 2 coagulant variable staphylococci, where both virulent and avirulent strains exist. Positive coagulase test results within the Staphylococcus genus are usually correlated to pathogenicity. S. hyicus is one of only 9 Staphylococcus species that can present as coagulase positive, which are the strains that are a cause for concern. But what is so interesting about this? Is it not like any other bacteria? Well, Staphylococcus hyicus has made a name for itself in veterinary microbiology and medicine. The agent, S. hyicus, has been accredited to cause exudative epidermitis in pigs, specifically young piglets. It has worldwide distribution, and farm settings are perfect breeding grounds due to the sheer volume of readily available hosts. Staphylococcus hyicus infections may occur in older pigs, although rare, considering immunity improves with age. S. hyicus may also infect birds, but more importantly, can cause infectious mastitis. Although it is usually not the causative agent, it is a condition affecting the mammary glands in cows, which can cause serious issues in the dairy industry due to abnormal milk production. A fascinating, in-depth article written by Peter R. Davies from the University of Minnesota further details the symptoms, transmission, and characteristics of exudative epidermitis from Staphylococcus hyicus. Transmission of S. hyicus typically occurs from a sow who has a vaginal Staphylococcus hyicus infection that is then ingested by suckling piglets. Suckling or young piglets comprise the majority of all and severe cases, with further transmission occurring with piglets at weaning when they are mixed together amongst other litters. Suckling-related transmission usually results in symptoms appearing in the facial region and orifices, and these hosts face an 80% morbidity rate that significantly drops after weaning, along with a roughly 5% mortality reported by the United Kingdom NADIS. The common name for exudative epidermitis, greasy pig disease, refers to the symptomatic oily secretion from blood vessels into nearby tissues, originating from red to brown lesions and dermal inflammation that are symptoms typically found on the legs, face, or anywhere on the body where skin is broken. It is crucial for infection that the skin is damaged, because S. hyicus has not been found to be able to infect through intact epidermis. Legs and feet are almost always affected, as the heels and coronary band are susceptible to cracking and dirt accumulation. Depression, loss of appetite, and dehydration are frequently observed in progressed cases, besides noticeable skin redness due to the virulent bacteria's exfoliative toxins, which target epidermal cells. It reduces the stickiness of the bonds connecting keratinocytes, which forms the outer barrier layer of the skin, and then degrades the proteins in the skin by breaking peptide bonds. Avirulent strains that are coagulase negative lack exfoliating toxins, allowing them to accumulate on epidermis without causing disease. Staphylococcus hyicus has been frequently mistaken for the more widely known Staphylococcus aureus, specifically in human medicine. When a 49-year-old male farmer with sepsis and cellulitis of the left foot was admitted to the University Hospital Bern, investigation identified edematous erythema, with procedural pinpricks resulting in pus exuding from the sites. Antimicrobial treatment was utilized with ceftriaxone, a class of cephalosporin antibiotics that act on bacterial cells by not only inhibiting growth, but also killing the cells as well. Biopsy testing revealed catalase and coagulase positive results, as well as white, non-hemolytic colony formation on Columbia sheep blood agar. This ultimately led to a Staphylococcus aureus identification, although characteristics such as a rapid symptom improvement in just 24 hours after antimicrobial implementation and unpigmented colonies for a typically golden yellow S. aureus did not exactly line up. So, further testing, including 16S ribosomal RNA sequencing, was undergone and revealed a 100% sequence identity with strain ATCC11249, GenBank accession number D83368, which is an exact match for Staphylococcus hyicus. Conclusively, the infection was decided to have been transmitted via zoonotic spread after the man claimed to have been in recent contact with piglets on his farm. While Staphylococcus hyicus has not been involved in any revolutionary news as of late, it should be noted that there are many recent tests to decipher the antimicrobial resistance of the bacteria, as treatment of swine is extremely important as to limit herd health deterioration. Further, the importance of the identification of S. hyicus as the cause of exudative epidermitis is impressive on its own, considering the disease was recorded some 150 years ago, while S. hyicus was only identified as a staphylococcus in 1965. Thank you for your attention, and I hope you learned something about the incredible Staphylococcus hyicus.